Hello everyone, in this video I'm going to be talking about something called cell fractionation or ultracentrifugation as it's called. The ultra refers to the size of what we're looking for, and the centrifuge is the machine that will allow us to do this. Cell fractionation essentially is a technique that allows us to separate out and look more closely at the organelles, the smaller structures found within a cell. Now, if you use an electron microscope, you can see the finer detail of the cell, but to separate out the organelles, you'd have to follow this process, and it's really straightforward. So we're just going to draw a very just basic cell. So we've got here the cell membrane, the cytoplasm inside. Let's say we've got the nucleus, the powerhouse of the cell where all the DNA is. Just here, some ribosomes we'll put in, a mitochondria, some endoplasmic reticulum. I'm just drawing very small, simple sketch because what I want to do in this is extract out one organelle. We're going to extract out the nucleus from a cell. So I want to keep that in mind. We're taking out an organelle, a smaller component of a cell, to examine. Now, the first stage is something called homogenization. So this is the first stage, homogenization. Now, what that involves is basically breaking up the cells. Now, we could vibrate the cells, that could do it, or even grind the cells up in a blender. So what we're trying to do is break up the plasma membrane and release all of those organelles into a solution. All of these parts, so the nucleus, the mitochondria, we're trying to release them into a solution. Now what you could have essentially is, so you've got a beaker here, let's say we've cut up a tissue, we've got it in this beaker. What we need to do is keep our sample in a solution that is cold, isotonic and buffered. I'm just going to explain what those three mean. So we'll homogenize and we break up the cells, and then we keep that solution in an environment that is cold, isotonic and buffered. So first of all, cold. The reason why it needs to be cold is to reduce the activity of enzymes, because enzymes could break down the organelles. So we need to make sure that their activity is reduced. So that's explaining the cold. Isotonic solution. Well, isotonic refers to the concentration of chemicals is the same in the solution as in the cells being broken down. And we're thinking specifically about water. So the water content of the solution is the same with the cells breaking down. Otherwise, organelles could burst or shrivel if water moves in or out by osmosis. So if there's more water in the solution that the tissue is kept in, water will move into the cell via osmosis and they could burst, or water, on the contrary, could move out. So we need to make sure it's isotonic or water balanced. And the final thing is buffered. It's important that the solution is buffered in terms of pH, because if there is a varying pH between the cells that you're looking at and the solution, then that can actually damage the cells, especially in an acidic environment. That excess of hydrogen plus ions that you have in acids can disrupt our cells. So we want a cold, isotonic, buffered solution. So our first stage is homogenization. Okay, the second thing we're going to do is filter. We're going to filter what is called our homogenate, or what is left over essentially, and we can filter them. We filter this homogenized cell solution through a gauze to separate any large cell debris or tissue debris, like connective tissue, for example, from the organelles. The organelles are much, much smaller than the debris, so they will pass through the gauze. So we filter to remove anything we're not wanting to look at. So we just have the organelles that we can focus on. Then the third and final stage is something called ultra-centrifugation. That's ultra-centrifugation. And what we do is we take that homogenized tissue and we spin it in a machine called a centrifuge at a low speed for roughly about 10 minutes or so. So what we have, if we just draw a little picture just to show, in fact. So let's take 2 chest tubes here, for example. Let's say there's, inside we've got our homogenized cell sample. And this machine will essentially just spin these tubes around. And after filtration, what you've been left with is a solution containing a mixture of all the organelles, and this spinning will separate the organelles due to size. So the heaviest organelles, like the nuclei, actually get flung to the bottom of the tube, forming a thick sediment, or a pellet as it's sometimes called, and the rest of the organelles stay suspended in the fluid above. Once you've drained off this kind of fluid above, you can extract this kind of pellet that you want and then spin again, and by this method, essentially by separating out heaviest first, you can extract all the individual organelles. So let's just shrink this for a moment and just explain what we're talking about. So let's say we spin these tubes. Now the heaviest of all the organelles is the nuclei. So when we'll have a look after 10 minutes, we'll find a portion of fluid, and at the bottom this what's called sediment. Sediment at the bottom, and the fluid that actually remains we give a special name. It's called the supernatant. So we spin, and because the nuclei are heavy, they drop to the bottom of the tube first and form this sediment. And then what we do is just extract this sediment. So we take out that sediment. So we're just left with the supernatant. And then we take another tube and we spin that again. And then we'll be left with a second sediment. And once again, there'll be some of the fluid remaining, the solution remaining, and that will form a second supernatant. And then we just continue, we just keep spinning. We drain off the supernatant each time, pour it into another tube and spin that. And the process is repeated at higher and higher speeds until all the organelles are separated out. So the nuclei are actually the heaviest. So right at the beginning I said I want to get the nucleus, we'll get that first. Mitochondria would come next. Then we'll be able to get lysosomes, then the endoplasmic reticulum. And then the small dots that are during the cell, those ribosomes, we'll be able to get those last. So this is ultimately cell fractionation. It's the method by which we can separate out the organelles and view them easily. So part 1, homogenization. We break that tissue up and can keep it in a cold isotonic buffered solution. Stage 2, we filter to remove any cell debris and leave just behind a solution with our organelles. And stage 3, ultracentrifugation. We spin and rely on the fact that the organelles have different weights to separate them out one by one. Okay, hope all that helps.